Hello everyone, and welcome to this audio podcast. This audio podcast, as part of the audio podcast series, the Storm Mode Isomer, and this audio podcast as part of the Miliplex. This audio podcast is part of the Mayhem and Twister, and this audio podcast as part of the Storm and the Storm Robotics production. In this audio podcast, looking at an overview of works around the science. A science student based on one of the had been based at one of the academic institutions in amongst the southwest United Kingdom, and one of the academic institutions in the southwest United Kingdom, and where this student this science student actually did did a, an over did actually compose some very sort of intricate some intricate detailed context in around looking at Oxford nanopore sequencing technology involvement in the genomic domain involvement in the genomic domain and its impact in the biomedical science field. So this science student Heidi Stefan, who had produced some fantastic works in around the sciences, and in on this audio podcast, under Storm Robotic production will be sort of an overview, and there will be citations or references in on social media, where listeners and viewers can look to refer to and check out some more detailed more detailed references and citations where to in around the theme of this particular audio podcast. So this audio podcast is an intro, and there is a synopsis. So do feel free to check out the synopsis in around this audio podcast. But to begin with, some intro in around this. So next generation sequencing, or NGS, is sort of a mixture of genomic technology capable of analyzing DNA and RNA to provide clinical information into various areas like molecular genetics, epidemiology, variant identification, genotyping, and disease and sort of disease relating or regarding research. Um, the first generation platform, or FGS, are broadly known as Sanger sequencing. Sanger sequencing methodology developed in around the 19 in the late in around the late 1980s, and was the start of the modernization of sequencing technologies, around the sequencing of technologies. Third generation sequencing, for example, TGS, has shown a sort of reduction has shown a reduction in sort of in some of the sort of in some aspects of the primitive technology in line to sample throughput, and in line to the sort of genomics versus the genomics versus the first generation elements. Oxford Nanopore sequencing technology, or as the students had abbreviated as ONT, is a TGS platform broadly used or utilized in genomics, where it reveals information on entire genomic structures, functions, and variability through DNA and RNA. Research into the genomic area can be used or utilized in diagnostics and predictions of different analyses and sort of forecast in some context in around different conditions, preventative sort of pathogenic elements and elements of personalized polyform and personalized sort of therapeutics, and also also in addition to a variety of pharmacotherapeutics, which can potentially positively advantage patient outcomes. And potentially advantage patient outcomes. So that draws this audio podcast, this intro audio podcast, to a close. This audio podcast as part of the Storm Mode and Isomer. This audio podcast as part of the Mayhem and Twister. This audio podcast as part of the Storm and a Storm Lobostics production.